Hello, good afternoon, and welcome to Nature Live Online from the Natural History Museum. I'm Khalil Thurloway. If you're new to Nature Live, this is a series where we have conversations with researchers from around the museum and beyond, telling interesting stories from their work in all sorts of fields, from dinosaurs to DNA, volcanoes to vampire squid, and much, much more. Today, we're going to be talking about how we can use technology to look at patterns in the natural world and see how all these living organisms are connected in their evolutionary relationships. To help us get the measure of this topic, I've got Anjali Goswami with me, a research leader at the Natural History Museum. Hi, Anjali. Thanks for joining.
Hi.
So before we get into the meat and bones of what we're talking about, why don't you give our audience a little bit of an introduction to who you are and what you do at the museum?
Okay, great. So I am a research leader in life sciences, but I'm a paleontologist by training, and I really work at the interface of deep time and the recent, incorporating data from fossils all the way through to embryos to try to understand the different factors that really shape the diversity of life on the planet today.
And if you're looking at kind of the diversity of life in all its richness, you must need a pretty big team of people working alongside you.
That's true. So in my group right now, we've got people working on everything from fishes to salamanders to dinosaurs and birds and all kinds of mammals. And we even have new projects that are focused on looking at insects and how insects evolve their incredible diversity. And so, yeah, we have quite a big group of people who really focus on different groups using, in some ways, similar methods to try and get a really comprehensive understanding of all the different things that really shape life on Earth. And so this is my team. The last time we were all in a room together, or at the museum together, and it's a really great way of working because these are really hard kind of methods and things to crack, and so we work really collaboratively to try and figure this out.
I think that's one of the wonderful things about science nowadays. It's becoming more and more and more collaborative, and needing all these different skills and all these different people to come to these kind of developments in research, rather than it being kind of one person locked away in their lab for ages.
Yeah, absolutely. It's definitely because these are really, because we're really at the frontier of science in many ways, with bringing all these new technologies and applying them to, in some cases, specimens that have been around for hundreds of years. It really helps to have lots of people to feed in new ideas and try different things out as we push forward.
And before we go on, there's one thing I forgot to mention at the top of the show, actually. For our audience at home, we want this to be a conversation, and we really love hearing from you as well. So if you've got any questions or comments, do pop them in the comments section down below, and we'll try and get to as many as we can throughout the show. Both Anjali and I are looking forward to reading some of those. But back to your research, Anjali. So if you're looking at the massive kind of breadth of evolutionary relationships across all sorts of parts of the natural world, and you've got this big team, how do you actually go about getting this information and analyzing it?
Okay, well, so what we're really trying to do is understand the diversity of life on the planet, how it's evolved now, how it's evolved in the deep past. And when people talk about diversity, there's lots of different things that people can think about. A lot of times when people talk about diversity, especially at a really large scale, over hundreds of millions of years or across the whole planet, they are often using a measure of species numbers. So how many species are there in different groups? But what we do in my group is actually a little bit different. We think about the diversity of form and trying to understand how different species look and the variety of different species in terms of how they look and what they do. And so to do that, it's really great on the one hand because we can bring a bunch of the data from the fossil record into these sorts of analyses. And this is really important because, of course, the vast majority of life that's ever lived is extinct. And so we want to make sure we're not just explaining how we got the incredible diversity of life that we have today, but also some of the really crazy forms that lived in the past as well. And so to do that, we have to gather lots of data. And so what we often do is we travel around the world using a lot of new technologies to put together 3D scans of different organisms. And so we do that using things like laser scanners and CT scanners where possible. And so in these images here, you can see that's me in Brazil. And that's one of my former PhD students in the collections here at the Natural History Museum using 2 of our laser scanners. And what we do with these is we scan the surfaces of the bones. We usually study the skulls, and we do that to capture the shape of the surface, and then we can analyze those using lots of 3D methods.
Why look at the skulls?
Oh, so skulls is something that we look at because skulls are incredibly interesting and incredibly complex. Skulls have lots of different functions. So, for example, they're obviously the key place where you feed and where you gather all the energy that you need to live. They also house all of our sensory or most of our sensory organs, so things like your eyes, your nose, your ears. They also, of course, hold the brain, and so it's a big part of what the skull does is support the brain and protect the brain, where you process all that sensory input. And in fact, a lot of organisms also use their skulls for lots of other things. So here's a 3D print of a dinosaur, Diabloceratops, and it has these crazy structures that it would have used for defense or for fighting, or potentially for display to other members of its own species or to other species. And so there's lots of different crazy functions that the skull does. And that means by studying the skull, we can actually reconstruct a lot about an animal's life, even if they lived hundreds of millions of years ago.
It's amazing in a skeleton with so many bones and so many different features to look at. In one bone or set of bones in the skull, you can learn so much about its lifestyle and how it would have interacted with the environment around it. So you showed us those images of you and Carla scanning those skulls, but is it tricky to get a kind of a complete scan, because skulls are very kind of irregular shapes and there's lots of nooks and crannies and stuff?
It can be really tricky. So in the ideal world, right, we have these skulls that we can move around and get from all different angles. But a lot of times, actually, there are things like this where they're actually still embedded in the rock that they were found in. Maybe you can only get half of the skull, then you have to use methods like mirroring them over. So this is a dinosaur that's in the museum in Drumheller in Canada. But it's a beautiful specimen, but we can only get, maybe 3/4 of it. And so you work with whatever you have access to. And we need really nice specimens in order to use this kind of approach, or we have to do this thing called retrodeforming them, where we get our 3D images and then we have to reconstruct them to get rid of any kind of damage that might have happened. And that also means that, that's just position and kind of where it's embedded, but size is also a massive issue. So some of the species in our data sets include things like Hope, the blue whale that's on display at the museum. And so being able to scan those really huge specimens also takes a lot of time and a lot of effort. Really tricky, but well worth it.
Is it easier or harder to scan a very, very large skull, like Hope the whale, or a really, really small one, like a hummingbird or something?
I think definitely really big is harder, because with the really small specimens, what we do is actually, rather than using our laser scanners, we use CT scanners. So these are these 3D X-rays. If you go to the hospital, you might have to get a CT scan. We have a couple of these at the Natural History Museums specifically for our specimens. And they're a really fantastic way of getting really, really high detailed images of even really small specimens. So we can do things that are, even just a couple of millimeters in size. We can get nice, really high resolution scans of those things. So definitely big is much worse than small.
So, but once you've got this scan of the skull, how do you get actual data and information out of it? Because you could just look at these scans of different skulls and kind of eyeball it and look for differences, but you take a more quantitative, a more numbers-driven approach, don't you?
That's completely true. And so, for centuries, really, people have been looking at skulls in what we'd call a kind of descriptive or qualitative way and picking out some of the big differences. But when you do that, you're kind of biasing your results by what you already can pick out of what you already think. But what we do is use an approach that is called morphometrics, where we're basically measuring the shape of the skull. Now, you could do this by things like measuring how long the skull is, how high it is. But what we do is we use these things called 3D coordinates, these little 3D points that we pick out on our different skulls, and use that to describe quantitatively the shape of the skulls using these different points. And we can try and find the same points on all the skulls in our data set and look at how the positions of those points have moved. Now, the problem is, if you're looking at really, really diverse skulls, like in this slide here, we've got birds, we've got crocodiles, we've got dinosaurs. They're pretty different in how their skulls are put together. They're pretty different in what bones you can pick out. even though they all are actually fairly closely related. And so what we do is we take take the points that we can pick out, and we use them to basically anchor a grid of much denser points. And we can lay those points out across the skull and then really compare, like capture the shape of that skull in really, really high detail, and then compare how the different parts of the skull have evolved.
So I guess it helps you to get past the kind of the big differences between these skulls can maybe confuse the kind of the human eye, the qualitative approach, but your approach kind of teases out that aspect and allows us to kind of just empirically look at what the kind of the analogous, the equivalent bones are on these different skulls.
Exactly.
And we've had a question from one of our viewers on YouTube who's asked, do you have to take into account the like the geology of the area where the fossil might have been found, or any of the factors that might have deformed or influenced the shape of the skull kind of in the ground?
Yeah, absolutely. I mean, that's really maybe the biggest limitation for us when we're gathering our data sets, is that a lot of amazing fossil skulls are smashed. They're either flattened or they're completely destroyed. And so we can do a couple different things. So we work with lots of collaborators around the world who are experts in reconstructing skulls in 3D from either their individual bones, or just if they're a little bit squashed, you can kind of digitally reconstruct what it should have looked like. And then you could even print it in 3D, so you have a real representation of something that actually, when you look at the actual specimen, is pretty flat. And so that's something that we do, and then we're trying to do increasingly to really build in some of those important fossils that are not in perfect condition. The other really fun thing that we can do is that we can use those data to guess what the ancestors would have looked like, and then try and match them up with the fossils that we find. And I think we have an example of that a bit later, but that's definitely a huge problem for us, that we have various digital techniques to try and get around.
So I guess it becomes like a kind of 3D digital jigsaw of this damaged skull.
Absolutely.
And just before we move on as well, we've got a question from another of our viewers on YouTube who's asked, do you have a favorite fossil skull that you've scanned, or a favorite organism skull?
Ooh, that's a good question. Do I have just one favorite? I feel like they're all my favorites in different ways.
Don't worry, we won't tell the other ones that you don't pick.
I think some of my favorite animals are actually some of the really crazy South American animals that have gone extinct. So you have things like these giant ground sloths, which look so crazy, and they're so different from the sloths that we have around today. But they were around only, they went extinct not even around 5,000 years ago. But if you just think that even that recently, they were so different from what's around today. So I love the South American mammals in particular because they're really weird. But I think my absolute favorite actually has to be, and actually I realize it's on my necklace right here, is this critter here. It's a Thylacus milis. It's a marsupial, so related to things like opossums and kangaroos, but it actually is a saber tooth. So it looks a lot like a saber-toothed cat, but it's completely separate. It's evolved from the kind of the pouched mammals, and it has these canines. Its canines actually grow forever, unlike the saber-toothed cat. So I think it's an even better version of a saber-toothed cat. So that's
even more saber-toothed.
Even more saber-toothed.
I find those types of what we call convergent or parallel evolution stories really, really fascinating. When you have totally different, separate evolutionary paths that have ended up with the same solution to a similar problem. Like in Madagascar, you have that whole group of tenrecs, which is a kind of a whole group of mammals that have evolved to fill lots of niches that mammals that we're familiar with in Europe have filled, and they look very, very similar. But they're totally unrelated.
Yeah, and you see the same thing in Australia with the marsupials in Australia evolving things that are very similar to the placental mammals, the group that we're part of. So you have like a marsupial wolf that looks really, really similar to a placental wolf. I mean, it's quite hard to tell their skulls apart, even though they're separated by 150 million years of evolution. You get marsupial moles, you get all kinds of things that really, convergence is really rampant. It's one of the really interesting and fun things, I think, if you look at the history of life.
I mean, I guess, there are certain designs that just tend to work in certain situations.
Yeah, absolutely. Good solution.
Sorry, we've got one more question that I think will lead us really nicely into continuing to talk about what we can do with this data. So Sophie has asked on YouTube, how do you choose where to put all those landmarking points on those skulls?
Oh, it's a great question. It's a really, really hard thing to figure out. And in fact, one of the nice things about having these 3D scans is that we can keep changing our minds about where we want to put them. And so, like, actually, when we were first figuring out how to do this project, we went through kind of 3 different what we call templates, or 3 different approaches before we figured out one that really worked well. But so what you do is you look at points that you can pick out on all the skulls. So things like where 2 different bones meet, or the tip of the rostrum and the tip of the snout, where the teeth end. So things that you can pick out really consistently. And then what we do is we take those points that we know we can pick out and we connect them with curves. And then within those, we use those curves to define basically the outline of the individual bones. And then we use those curves to anchor these surface points. And so we start from what we are 100% sure of, and we use that as an anchor for all of the other points.
And what can we use this data for? What can we really learn from the information that we get once we've got these comparable points between these different skulls?
Yeah. So what we can do then is, once we have this really good description of the shape of the skull, then we can try to reconstruct how it evolved in a really, really nice level of detail. So what we do is we take our understanding of how different species are related to each other, and we can do things like reconstruct, okay, along this branch of the family tree, the skull changed from this, and what it looks like in an ancestor, to kind of this version that it looked like in its descendant. And we can reconstruct how fast that happened. We can reconstruct if kind of similar changes or similar speeds of changes happen at the same time. So for example, we can look at past periods of global warming and say, okay, during this, we know that during like this, 1 million year period, you had this really rapid change in temperature. Now, if we look across all these different organisms, do we see that there was big changes in how quickly species were evolving? Or can we see that certain kinds of species evolved really fast, or evolved well, or evolved into something that could survive those changes? And did other kinds of species go extinct? So we can start to look at how evolution happens, reflects things like big environmental changes. We can look at if different kinds of organisms evolved in similar ways, like if do all the things that eat plants do well under certain scenarios, and do all the things that eat meat to show the same pattern. We can start to figure out which of these factors is really critical for shaping the evolution of life, and which of them is really maybe not so important, or which ones actually limit how well species can evolve or can't evolve. And so we can do lots of those kinds of analyses to really try and figure out what are the most important factors that really shape the evolution of diversity, and also which ones are really important for understanding how species respond to large-scale changes like mass extinctions or big environmental changes.
Well, on the subject of big environmental changes and mass extinctions, you recently did a study on looking at the rates of evolution in birds and dinosaurs. Let's talk about that a little bit, because as some of our viewers might already know, this might be new to some of you, birds are essentially dinosaurs. Birds are the descendants of a group of dinosaurs. And so by looking at how dinosaur skulls evolved and how bird skulls evolved, you've been looking at the rates of evolution and how what that says about the importance of different features of their skull, right?
That's right, yeah. And so the bird dinosaur question is obviously a really interesting one, because we have this incredibly successful group today. We have birds. There's 10,000 species of birds around today. They're a really dominant group on Earth at the moment. But of course, they're so different from their ancestors, and we know that birds evolved from theropod dinosaurs, and we know that birds today are quite different than what we think of as dinosaurs, right? Dinosaurs were these, in many cases, gigantic beasts that completely dominated the planet. They have this huge diversity of forms. And so we can see some of the examples up there, and I've got some of those with me right here. So you can see, they have these fantastic, really ornate skull structures or skull ornaments. You've got really kind of weird features like that are evolving on the different parts of their skull. Whereas when we look at birds today, we have this immense diversity of things like their beaks. So you can see all the ones on the right there are birds, and they have this huge diversity of beak forms and this huge diversity of ecologies, but they're very different in how they have evolved compared to their dinosaur ancestors. And so what we wanted to know is, once you can figure out a way to actually compare these so this incredible diversity of skull shapes. Do birds really evolve faster than other dinosaurs? Is that why we have so many species of birds around today? 10,000 species of birds around today, which, by some estimates is maybe 10 times as many as there were as dinosaurs that we know about, at least in the Mesozoic. And so do we see this explosion of diversity when birds evolve? Which is something that people often think about, that, when the other dinosaurs went extinct, birds as well as mammals really show this big explosion of diversity. And so what we'd expect to see is a big increase in rates of evolution. What we actually found when we measured out all the different shapes and we reconstructed using their family tree, where on the family tree do you see fast evolution happening? The fastest evolution is actually not in the birds at all. It's actually in all the other dinosaurs, especially some of these groups that are kind of further away from other dinosaurs, birds, things like, the big horned dinosaurs and things like that. And so that's really interesting because it's totally the opposite of what we expected. We actually were able to measure, and on average, dinosaurs evolve something like 1.5 to 3 times as fast as birds do today, which is an incredible slowdown actually in evolution when you get to birds. And so then we were thinking about, well, why is that? And actually, when you think about it, it kind of makes sense, even though it's the opposite of what we would expect. And actually, the funnest part, I think, of science is not when you prove something that you thought, but actually when you realize you were completely wrong. Once you have the data, you completely have to change your perspective on what you thought about, the evolution of life.
Yeah, absolutely.
And so when you think about it, bird skulls are all about, they've got these big brains and these big beaks, but actually not that much else. Whereas dinosaurs were doing lots of crazy things with their skulls. They have these big ornaments. They have all kinds of diverse feeding structures. They've got also chewing and things like that birds, they don't really do. They have all these crazy teeth, and birds, of course, don't have teeth. And so, but they didn't have really big brains, actually. Most of their skull is about, how to attach their jaws up and how to have all these weird ornaments. So I think actually it's just that birds kind of only do one thing with their head, whereas dinosaurs just do a lot of different things with their head that birds today do more with, like, their feathers and other things. And so that was a really surprising result. I think a really, really interesting switch in our perspective on birds. They're still amazing, but not as amazing as I thought they were.
So you said that there's like a massive diversity of birds now compared to the diversity of forms that we saw early on. Where did that diversity come in? Would it have come in during the kind of pre-extinction period when there were lots of dinosaurs, and then they kind of became very diverse, and then sorry, I'm having trouble kind of unpicking that.
Okay, well, so I think one of the other issues is that we're talking about 2 different kinds of diversity, right? So birds have lots of species, but if you actually look at the variety of forms, a lot of what we think about in the really amazing variation in birds is actually not about their skulls. It's actually more about their feathers and things like that. So actually, if you take all the skin off, they're not so diverse actually. But there's lots of species of them, and they kind of separate their species in different ways using things like their feathers and coloration or song and things like that. So on the one hand, birds have lots of species, but in terms of the variety of forms, I think the non-bird dinosaurs actually show a greater variety of forms. These big sauropods and these big T-Rexes and these big, horned dinosaurs and everything. And then the other part of your question in terms of when does this happen? So we had some fossils from kind of the early birds. What we're working on right now actually is trying to fill that in with more fossils of those early bird forms. But actually, a lot of the bird kind of radiation, as we call it, or the big diversification of birds, happens after the other dinosaurs go extinct. And a lot of the Cretaceous birds that were around when the other dinosaurs were around, they also go extinct around in that mass extinction. And the ones that are around today are very much a kind of post-extinction explosion of forms.
And we've actually got a question from one of our viewers. Thank you for clearing that up, by the way. That's really helpful. We've got a question from one of our viewers on this kind of similar topic, asking why birds, you don't really see them with teeth, but a lot of their ancestors, their direct ancestors, were extremely toothy. Do we know how and when and why that transition happened?
Yeah, well, why is always a difficult question. You can think that it probably had something to do with diet. So we do see tooth loss happening a lot in different groups of organisms, usually related to changing their diet towards something that's maybe more towards insect eating or something along those lines, moving away from the kind of really chewy or bitey kind of diets. And once you lose a structure, it's really, really hard to re-evolve it. So this is kind of a classic trend in or law of sorts in evolution, that losing a structure is a lot easier than gaining or regaining a structure. So in terms of why birds don't have teeth today, a lot of it has to do with the fact that they lost their teeth, and getting new like re-evolving those teeth is really difficult. Now you can actually do it in a lab by tinkering with their genes. And actually, kind of interestingly, and one of the new members of my lab is going to be working on this for her PhD. There are some groups of birds that re-evolve teeth, actually, so kind of develop pseudo teeth to try and regain some of that function. But actually, regaining the teeth like what we have, or what a lot of the dinosaurs would have had, when you've already gone so far as to lose your teeth and really kind of cover it up with a beak made out of keratin, I think is a really, really tricky thing to do developmentally, because you kind of shut off the genes that do that.
Wow, that's actually a lot of food for thought, because sometimes you do see, for example, little serrations on a goose's beak. Would they be do they just look like teeth, or are they kind of the remains of actual teeth?
No, they don't look. They're not the remains of teeth. They're kind of a separate evolution of a tooth-like structure. So it's sort of convergence in that way. But
Different tool for the same problem.
Yeah, exactly.
So this is really fascinating, and you're in this example. These examples we've just been talking about, you're comparing some quite closely related organisms, birds and dinosaurs. But can we kind of broaden this analysis and comparison out to look at bigger trends in evolution?
Yeah, absolutely. So that study is part of a much bigger project in my group, where we're trying to do this across, well, originally we were looking across all land vertebrates, and now we're working on fishes as well. And so what we've done is taken the same approach. And on this slide here, you can see we've got birds up top and dinosaurs right below them. But then we have lizards, snakes down in the middle at the bottom, and then different groups of amphibians. So we have cecilians, which are these limbless amphibians. We've got frogs and salamanders, and we have a bunch of data for mammals that we're analyzing right now.
So going anti-clockwise from the top, we've got birds, dinosaurs, lizards, snakes, and then amphibians?
Yeah, that's right. Cecilians,
Awesome.
frogs, and salamanders.
Great.
And so what we're trying to do is do this same kind of analysis across all of these different groups, including essentially all of the vertebrates ultimately, and try and figure out how their patterns of skull evolution differ, and what are the common factors across all of these groups. So for example, do things that lay eggs evolve faster or slower than things that give live birth? Do things that go through metamorphosis evolve faster than things that don't? Do things that eat plants evolve faster or slower than things that eat animals? And try and figure out if there's general rules really governing the evolution of vertebrate diversity. And in fact, we're starting to work on this, as I mentioned, in insects also, trying to understand if there's general rules for why that can explain why insects are so diverse. And so what we can do with this, which I think is really cool once we have this kind of data, is so on these skulls here, you can see the landmarks. Those are those individual points that we put on all the skulls. But in this case, they're colored by how fast those different parts of the skull evolve in these different groups. So for example, if you look at the bird, you can see that the tip of the beak is really red. So red means that it's evolving really fast. And so in birds, if you look at birds today, a lot of the evolution, the biggest way that birds differ is really in the shape of their beak. So if you think about things that eat fish versus things that eat nectar or eat seeds, you can see that variation is really about the shape of their beak. But if we look at actually most of the other kinds of vertebrates that we've looked at, well, it's actually pretty unusual. And for all the rest of them, kind of the reddest areas are usually kind of back where their jaw meets their skull. And so that's really about what's controlling how they bite and how they chew. And so that's actually quite interesting. So you see this shift from within the dinosaurs, going from something that's kind of a more typical pattern that we see in snakes and lizards and amphibians to something that we see in birds, where you start to see more of the diversity being focused towards the tip of their snout as opposed to how they bite. And that has to do with things like jaw muscles and probably also the size of the brain. And we see something similar happening in mammals too. So it's quite interesting to think about, well, what are the common features then that's kind of driving those patterns?
Either way, it sounds like food might be one of the big features if it's the tip of the beak and it's the jaw of the other animals.
Absolutely. And it, in fact, could be reflecting really kind of fundamental developmental processes in genes and how there's trade-offs between different genes and trade-offs between different functions, and, different ways of feeding versus the size of your brain. So I think it's about how different groups are really emphasizing the different things that the skull does.
And this actually links really nicely into a question from one of our viewers, Robin, on YouTube, who's asked, with genetic data also becoming much more available and accessible in recent years, will we be able to study and connect how differences in bone structure relate to differences in the genes of an organism?
Yeah, absolutely. So that's definitely where we're going, and we've been gathering some preliminary data to try and think about what different genes are really active in kind of this part of of the cell, and what genes are really active over here, and why those ones seem to be the most concentrated variation, or why are all the differences that we see really concentrated in those parts of the skull? And so we have a list of what we call candidate genes that we think might be really driving these patterns. And now what we want to do is really start to get comparative data on the genomes and maybe do some experimental work to really see how these patterns that we can see across all these species, now that we know where to look, we can start to look for the actual mechanisms, the genetic mechanisms that are driving those patterns.
And I guess as techniques and technology become more and more advanced, it becomes more and more possible to access genetic information from older and older samples as well. There are already specimens that we can get genetic material from that 10 years ago we would have thought it was impossible. So who knows where this will lead? That's
true. I don't think we'll quite get to the Jurassic Park level, but we can definitely some of the more recent extinct organisms, like like some of those animals that I was saying are some of my favorite things, the crazy things that are in South America that went extinct relatively recently. We can start getting genetic material out of them, which is cool because we actually don't really know where they fit into the tree of life. And so without those genetic data, we were kind of at a loss, but now we're starting to get some understanding because of that. And so I think there's, tons of new things that are going to become available over the next few years to really help us bridge, this really rich data that we have on genetics and genomes, and this new rich data that we have on what organisms look like.
Well, I'm looking at kind of the relationships and kind of evolutionary timelines of kind of not very closely related but sharing the same habitat organisms. We've got a really interesting question, I think, from one of our viewers from Hong Kong, who has said, it was asked, why do we know why the megalodon, the giant shark, prehistoric shark, died out, but the ancestors of modern whales didn't? Because they were both carnivorous, large ocean-going organisms. I guess it's harder to study megalodon with your kind of techniques because actually there's very little of megalodon that remains in specimens. It's mainly the jaws and teeth.
Yeah, that's a really good point, actually, because sharks are largely made of cartilage, right? A lot of what we have for sharks are just the teeth because bone and especially teeth are so much harder than cartilage. They actually just preserve much, much better. And so, yeah, that is a big limitation, actually. And that's one of the reasons why we focus on things like skulls and bony materials, because we actually can get good fossil material for a lot of them, whereas things like sharks are a little bit more difficult. That said, there is a great fossil record for sharks going back hundreds of millions of years, and some really, really fantastic fossils that show incredible diversity of form. But yeah, capturing that with these kinds of approaches is, I think, maybe a problem for the next project, because it is quite tricky. We're just starting to delve into fish right now, and because their bones are quite different and put together in quite a different way from the way the things that live on land are. It's a new set of challenges.
It's a new fun project. So it's someone else's PhD thesis. And Sophie has asked, do you think that you would get similar kind of trends coming out, or even kind of totally new and unexpected trends if you looked on other bits of the skeleton? So like hands or hips or something like that.
Yeah, that's a really good question. And so we focus mainly on the skull for the reasons I mentioned earlier, but we can look at other parts, so we've got data on the jaws, we've got some data on the limbs. We did a whole project recently where we looked at the entire skeleton and reconstructed its evolution across cats. And what's really interesting is that you're exactly right that you don't get the same results if you look at different parts of the skeleton, because different parts of the skeleton have different relationships to ecology. They have different relationships in terms of their development and how kind of limited they are because of how functionally, how changing them would maybe disrupt really key functions. So for example, if you look at the backbone actually evolves quite slowly relative to things like the skull and the limbs. It doesn't show as much kind of what we would call ecological signal as those other parts of the skeleton do. The limbs really reflect in many ways locomotion and how an animal is moving around. In some cases, it'll reflect diet. So if, for things like cats that actually might use their forelimbs to grapple, or things like birds that will use their hind limbs to capture prey, you see some of that signal, but they reflect different aspects of an animal's life. And so I think you do get differences if you're looking at the limbs versus the backbone versus the skull, because they are driven by different functions in many ways.
And also, I guess there's an element of how many winning designs or how many successful iterations or designs can you have on that particular part of the body. So, for example, there are all sorts of different beak shapes that can do different cool jobs, but how many times can you reshape a spinal column and it still do the job it needs to do?
Yeah, that's very true. Because the spinal column is also really tied to things like breathing structures and things like that, and protection of obviously all of the intestines and things. Yeah, I think it is just more limited in many ways than some of the other structures. So you do tend to find slower evolution in those ways. It also depends a little bit on the kind of organism. I think mammals like us tend to be actually pretty, we have a pretty kind of set way that our backbones are made. But if you look at things like reptiles and fishes, they show a lot more variation. And that probably reflects things like how we breathe, as well as possibly how we reproduce. So there's different things that can kind of both promote the evolution of these different structures, but there's also lots of different things that can limit how they can evolve.
And we've had a question on YouTube that I realized maybe we should have addressed right at the beginning of the show. But we've been talking a lot about fast and slow evolution and the evolutionary speed. And yeah, we've had one of our viewers asking, how do we define that? What do we mean by evolutionary speed?
Oh, that's a really great question. And there are different ways of defining it. But actually, probably the easiest way of describing it is if you take a branch of the family tree, and you look at what the ancestor on that branch and what the descendant looks like, and you could measure using the methods that we've been talking about, how different the ancestor and the descendant are from each other, and you can kind of divide that by how long the branch is. So has it been 5 million years of evolution in that branch, or has it been 20 million years? And then you can basically come up with, okay, per million years, you see this much change in this structure. And that's essentially what we mean. And so do we see more change on this branch per million years, or do we see more change on this branch? And that's basically how we're trying to compare how fast evolution is happening.
And so here with this, with what's called a cladogram, which is a kind of family tree of these organisms. If you can see for our viewers in the middle, you've got these kind of lines radiating out and branching out. These are just like in a human family tree. These are the places where different groups of organisms branch off from each other, and they share a common ancestor closer to the middle, which is earlier on, and they kind of branch out and become more diverse as you get to the outside, which is more recent.
Exactly.
So when Anjali's talking about distance and time, that's how we're looking at it in this situation.
And those kind of gray, the different levels of gray are the boundaries between the different kind of geological periods. So you can see the Triassic and the Jurassic and the Cretaceous, and that shows when those different groups would have diverged or split off from each other. And then so we can look at the time since they split off and estimate how fast they've been evolving, and then compare that across the different groups.
So in terms of kind of adaptations to the skulls, we've got a question from Megan on Facebook who's asking, I think, a question that's probably on a lot of people's minds. What's the weirdest skull you've ever seen?
I have a 3D print of it right in front of me. I would say it's this. And the reason this one I think is so strange, this is a theropod dinosaur. And what's weird about it is it's called City Potty, and I always get it backwards, actually. I can never quite tell the front or the back of the skull is. I think this is the front, but half the time I look at it, I get it wrong because it's so weird looking.
It's got so many holes. Where are the eyes?
Yeah. I'll probably get it wrong if I try to point out. I think they're here, but it just looks so symmetrical and so unusual compared to, even like something like this ceratopsian is quite normal looking. But then you have this thing, and it's always very confusing to me. So I think this is actually the weirdest skull I've ever seen.
Yeah, the City Party, you could hold it in almost any orientation and it would still look like a face.
Yeah. So this is the front. I'm pretty sure. Yes, this is the front over here. But it
looking
really,
for the dual process.
Looks like that could be the back easily. So I think this is definitely the one that I always find the most confusing to look at.
It's like a magic eye, but in 3D.
Yeah. Although to be fair, if you've ever seen a sperm whale skull, or really any of the big whale skulls, they are incredibly weird looking. They look like airplanes. So those are also, I think, one of the weirdest things that's ever evolved.
Sorry, that's the sound of all of our viewers Googling sperm whale skull. So I think we're getting towards the end of our conversation. So I think it might be nice to wrap things up with a kind of, so if there are any kind of budding comparative anatomists or paleontologists out there who have been absolutely fascinated by all this talk of skulls and scanning and evolutionary relationships and unpicking these kind of family trees, what can people do to kind of find out more, or maybe get into that field?
Yeah. So, I mean, one of the main things, obviously, is just getting out and learning more about nature and looking at animals and plants and just all the things that are around. Even in the middle of a city, there's fantastic places where you can explore nature. And so that's, one of the main things. And that's really what drew me into this field. There's also a lot of great resources online. I mean, if you just want to get your head around skulls and look at some of the amazing diversity of skulls, one thing that we do is that, as we're gathering all this crazy data, in these 3D scans for thousands of species, we put them online so that anybody can download them. So you can download, these skulls and print one out yourself if you have a 3D printer or access to one. And so if you go to phenome10k.org, that's where we upload all of our skulls as we publish them. And you can rotate them, look at them, and play around with them and get a sense for just this immense diversity.
Well, there goes my afternoon.
Yeah, exactly. It's really easy to get lost in a bunch of skulls, I think.
And so for our audience, I think we're going to be putting a link to that URL in the comment section, so you'll be able to see that a bit later. But yeah, so I'm really looking forward to spending my afternoon just looking at skulls. I think that's all we've got time for today, though. So thank you so much, Anjali. I've really enjoyed this and learned a lot. I love skulls in general, but talking about this kind of aspect of it is really, really fascinating.
Yeah, thank you. And thanks for all the great questions.
Oh, yeah, thank you. So thank you so much at home for joining us as well. I've really enjoyed myself. I hope you have as well. Anjali and I have both really enjoyed reading your questions and discussing them. So that's it for this edition of Nature Live Online. Do join us again this Friday at 12 and every Tuesday at 10. No, apologies, getting my days mixed up. The pandemic has done funny things to our calendars. Tuesdays at 12 and Fridays at 10:30 a.m. And the museum is open still, although we're under the Tier 2 restrictions. Make sure that you do check whether you're abiding to the regulations when you do come and book a visit. You can find out all the details on how we're trying to make sure that everyone has a fun and safe visit on our website. Until then, thank you so much. That was Anjali Goswami. This has been Nature Live Online, and I'm Khalil Thurloway. Thank you. Goodbye.